Okay, so I'm going to talk about DNA replication. So DNA replication is when we replicate DNA, we do it in a semi-conservative manner. This means that when we replicate the DNA, there are two copies that are produced. They contain one of the original strands and a new strand. The strands are complementary and anti-parallel to each other. Okay, so the things involved in DNA replications are the RNA primers. The RNA primers are made by RNA primase, and the Okazaki fragments are joined by DNA ligase. DNA polymerase is the enzyme that carries the daughter nucleotides, and the DNA helicase is the one that unwinds the double helix to open the replication fork.
Transcription. Transcription is the first step in gene expression in which information from a gene is used to construct a product, such as proteins. Transcription's main purpose is to make an RNA copy of a gene's DNA sequence. There are 3 stages in transcription: initiation, elongation, and termination. Initiation is where RNA polymers binds to a sequence of DNA called the promoter. Once bound, RNA polymers separates the DNA strands, providing the single-stranded template needed for transcription. Now, elongation is where one strand of DNA acts as a template for RNA polymers. The polymers build an RNA molecule out of complementary nucleides, which make a chain. The RNA transcript carries the same information as the non-template strand of DNA, but has a different base instead of thymine. Now, the last stage is termination. Here, sequences called terminators signal that the RNA transcript is done. Once they are transcribed, they cause the transcript to be released from the RNA polymers.
So inside of cells, many things happen, and they must all be described in different things. After my group has already described DNA replication and transcription, I, Andres Ornelas, will be describing translation. So when transcription is complete, the messenger RNA is passed through the nuclear envelope, entering the nucleus. The RNA is directed to the ribosome inside the nucleus, and after attaching to the ribosome, the process of translation begins. The job of this mRNA is to carry the gene's message out of the nucleus to a ribosome. Inside the ribosome, a molecular assembly line puts together a specific set of amino acids. The amino acids are brought from different parts of the cell, and their sequential arrangement determines the type of protein that is made. These proteins are essential for life.